Strange new species found in toxic lake could hold clues to life's origins. Scientists have discovered a new microbe species, Barroeca monosierra, in the highly inhospitable Mono Lake in California. This new species belongs to a group of single-celled organisms called Choanoflagellates, often referred to as choanos by the research team. Choanoflagellates are closely related to animals, and their colony-forming behavior might offer insights into the transition from single-celled to multicellular life. Barroeca monosierra is particularly notable because it was found thriving in an environment much saltier and more toxic than usual for its kind. Unlike other choanoflagellates, which are typically solitary or form smaller colonies, B. monosierra forms large clusters of up to 100 cells, 4 times larger than related species. These colonies also contain a hollow center filled with smaller communities of live bacteria, making B. monosierra one of the simplest organisms to maintain its own microbiome. The study, led by researchers from the US, UK, and Spain, offers several hypotheses about why these bacteria live within the colonies. The bacteria could be taking shelter from the toxic lake water, or they might be cultivated by the choanos as a future food source. Alternatively, a symbiotic relationship could exist, benefiting both the bacteria and the choanoflagellates. Choanoflagellates, such as B. monosierra, are of special interest to biologists because they are considered the closest living relatives of animals without being animals themselves. Their ability to form colonies is thought to mimic an early stage of multicellularity, potentially representing a transitional form between single-celled and multicellular organisms. The colonies of B. monosierra are structured similarly to a blastula, which is a hollow ball of cells that forms during the early stages of animal development. Mono Lake, where this new species was discovered, is a challenging environment for life. The lake, located east of the Sierra Nevada and near Yosemite National Park, is almost 3 times saltier than the Pacific Ocean and filled with various toxic chemicals, such as chlorides, carbonates, and sulfates. Due to these extreme conditions, the lake hosts very few life forms, including alkali flies, brine shrimp, and some species of tiny worms. However, the researchers were surprised to find large, thriving colonies of choanoflagellates in a water sample from the lake. Upon further examination, researchers found that the B. monosierra colonies have a unique structure. Unlike other choano colonies where cells cluster with their heads together, the B. monosierra cells are connected by an extracellular matrix made of protein and carbohydrates, forming a hollow center. DNA staining and RNA probing revealed the presence of bacteria in the center of these colonies. Further experiments using fluorescent amino acids showed these bacteria were alive, not merely remnants of previous meals. Interestingly, not all bacteria from Mono Lake are found within these colonies. Phylogenetic analysis demonstrated that only specific types of bacteria are present inside, suggesting a selective entry mechanism. Additional experiments using latex microspheres of similar size to bacteria showed that these did not end up in the colony centers, reinforcing the idea that the bacteria are actively chosen or allowed in. Nicole King, a cell biologist from the University of California, Berkeley, noted that this discovery marks the first time a choanoflagellate has been observed with a stable physical relationship with bacteria, potentially forming a symbiosis or a microbiome. Prior studies only showed choanos responding to bacterial molecules in the water or consuming bacteria, but nothing that suggested such a close, stable interaction. The discovery of B. monosierra opens up new possibilities for studying the interactions between bacteria and more complex organisms, potentially shedding light on how life made the leap from single-celled to multicellular forms. The researchers aim to use this species as a model to further explore these interactions, which could also help us better understand the origins of complex life. The findings were published in the scientific journal mBio.